Welcome, everyone. Today we have Dr. David Jin, President and CEO of Nasdaq-listed Avalon GloboCare, ticker symbol AVCO. Avalon GloboCare is a clinical-stage biotechnology company dedicated to develop and deliver innovative and transformative cellular technologies and therapeutics in the field of immuno-oncology. Their core technology platform is trimeric antigen receptor, or CAR-T therapy. Dr. Jin, we're excited to learn more. How are you doing today?
I'm doing fine. Thank you, Michael. Thank you for having me here today.
It's our pleasure, Dr. Jin. Very excited to learn more about CAR-T therapy and several other things that Avalon is working on. To get us kicked off and get some background, can you briefly explain to our audience what is CAR-T therapy?
Sure, Michael. As you just mentioned, Avalon's core technology platform involves the re-engineering of T cells with chimeric antigen receptor, or CAR, for potential treatment of cancer. As we know, T cells are the workhorses of our immune system to fight cancer. Over the last 10 years, CAR T cell therapy has been the biotech industry, has taken the biotech industry by storm, creating hope that it could usher in a new era of cancer treatment. A conventional treatment protocol about CAR T therapy, we have the T cells from the peripheral blood of a patient are isolated, then using a disarmed or harmless viral vector to deliver the CAR molecule into the T cells. This CAR molecule carries a molecular GPS system, recognizing the tumor cells very efficiently, as well as a tumor killing activity once it binds to the specific target on the cancer cells. Once reinfused back to the body of the patient, these CAR T cells get multiplied and stay in the body as living drugs. Theoretically, these CAR T cells has memory and stay in the body to suppress the recurrence of cancer.
Wonderful. Now, Dr. Jin, let's dive a little bit deeper into the subject. Just this last week, Avalon announced a collaboration with the University of Pittsburgh Medical Center to co-develop a new cancer immunotherapy involving Avalon's mRNA-based FlashCAR platform. Can you tell us more about this mRNA immune cell therapy? What are some details on that?
Sure. Probably, every one of you have heard about mRNA, especially now we have this COVID-19 mRNA vaccine by Moderna and Pfizer. At Avalon GloboCare, we take advantage of this powerful and versatile gene engineering technology to produce CAR T cells to fight against cancer. Avalon GloboCare has established our proprietary FlashCAR platform to make these mRNA-based CAR T cells to tackle cancer. Now, cancer cells has a signature feature. These are called the tumor-associated antigen on its surface that can trigger our immune system. With our FlashCAR technology platform, a designer mRNA or a messenger RNA, which provides the blueprint to create the CAR molecule and delivered, to the patient's T cells and turning the T cells into a biofactory, making the designer CAR molecules on the surface of these CAR T cells. You can imagine, as I mentioned before, each CAR molecule has two functions: a molecular GPS guiding system so that can mobilize the T cells to the tumor site, as well as a weapon that can bind to the signature tumor-associated antigen and trigger the immune-driven cancer-killing effect. So that's basically the nutshell of our FlashCAR system.
Okay, wonderful. And so let's speak a little bit to AVA011, the mRNA CAR-T candidate that you have in play. How is that more superior than conventional CAR-T therapy? Can you give us a little bit of background there?
So right now we have about 5 approved CAR-T products by the US FDA. These are the early generation of CAR-Ts. We know they are very powerful and can revolutionize our treatment options for cancer patients. Scientists, investors, and developers, they invariably agree that CAR-T provides a very powerful tool for cancer treatment. However, the current generation of CAR-T have faced several limitations and challenges. The first one is there's a high risk of adverse effects and toxicities, mainly that's called cytokine release syndrome or cytokine storm. Another one is a neurotoxicity called CRES, C-R-E-S, which is a CAR-T-related encephalopathy syndrome. For our AVA011, which is an mRNA derived CAR T cells, we put in a molecular safety switch in the mRNA construct such that once these mRNA CAR T cells infused into the cancer patient's body, if the patients develop an onset of these CRS or CRES syndrome, we can shut off all the CAR T cells that are already in the body by giving the patient an antidote, which is a small molecules that it's a drug that already approved by the FDA. So we believe, we have a game changer in terms of a superior safety profile. Another advantage for our AVA011 is that for the conventional CAR T cell therapy, it has a very long biomanufacturing time. Conventionally, it takes at least 14 days. We call it from vein to vein time, from isolating these T cells from a patient's body and then work in the lab and then give it the viral vector, expand them, and then finally, put it back to the patient's body. It takes about 14 days. And also it incurs a very high cost. Believe it or not, right now the average cost for a CAR T infusion is about $500,000 per dose. For our mRNA CAR T, we can achieve a very short biomanufacturing time. It takes only 1 to 2 days of manufacturing, and it just takes one step, what we call the electroporation, to deliver the designer mRNA into the patient's T cells without using a viral vector. So it much reduced the biomanufacturing time as well as the cost. Another advantage of our AVA011 is that it is a multiplex approach. Even we use T cells so that we can create an mRNA-based CAR T, but the mRNA actually contains some functional units which allow us to activate other immune system cell types, including natural killer cells, dendritic cells, macrophages. So the whole repertoire of the immune system will be activated to fight cancer after the patient receives our mRNA CAR T. Now, another important point is that right now all the conventional CAR T that are approved by the US FDA, these are all autologous approach, meaning that it's patient-derived and leads to a very high cost and very long manufacturing time. With our mRNA CAR T, we can generate allogeneic or universal off-the-shelf approach. So we have a super donor that who can give us the healthy T cells such that we can use a pharmaceutical approach to make off-the-shelf or universal mRNA-based CAR Ts. So we can store them in the freezer such that whenever we have a patient, suitable for these, CAR T therapy for their cancer, it's all readily available. So the AVA011 targets both CD19 and CD20 tumor antigens for the indication of the patients with relapse and refractory B-cell acute lymphoblastic leukemia and lymphoma. In the future, we will also design different mRNA-based CAR T with tumor antigen targeting solid tumors. And this is the holy grail in the whole CAR T therapy industry. Our mRNA-based FlashCAR platform is a game changer, which allow us to mitigate those challenges and limitations faced by the current approved CAR T products.
And Dr. Jin, as we kind of zoom out here and wrap things up as we talk about CAR T, can you quickly share with us the developmental timeline from that standpoint of this fascinating living drug? And when do you expect to see AVA-011 entering its first in-human clinical trial?
We are now collaborating with UPMC Hillman Cancer Center. And currently we are in this, we call it IND enabling stage, to generate clinical grade mRNA-based AVA-011 CAR T cells in preparation for our upcoming first-in-human clinical trial. We expect to have a better and safer CAR T immunotherapy available for our cancer patients during first quarter of 2022 in a first-in-human clinical trial setting.
Fantastic. Dr. David Jin, CEO of Avalon GloboCare, thank you so much for joining us and talking about CAR T and so many different applications and things you're going to be able to do with this kind of developments you're working on and all of the potential and growth for Avalon as well when you're discussing CAR T and all of the applications. Thank you again for joining us, Dr. David Jin.
Thank you, Michael. My pleasure.